Welcome everybody to the February 17th meeting of the IMAG-MSM Working Group on Multiscale Modeling and Viral Pandemics. We have a single talk scheduled for today, so I hope people will have a chance to ask some questions. As always, I need to remind you that this meeting is live streamed on YouTube and is recorded, so please keep that in mind as we go through the meeting. As always, you've seen this slide many, many times now, more than 50 times. Reinhard and myself are always available for your comments and suggestions. Jim Sluka as well, and you've been getting emails and correspondence from Lorenzo, who has been doing an amazing job on trying to recruit speakers and organize the activities of the working group. We appreciate that very much. We have our usual communication channels. Please help us work on them. Especially we have our YouTube channel. Help us annotate that and distribute it. Make sure that people know about the great resource of talks that we have here. Help us retweet our events. MSM Viral tweet helps us get the word out. Thanks very much for that. We have time for a few brief announcements. I know there's at least one on the schedule, and I'll first start by saying that we have a steering committee meeting tomorrow morning at 11 a.m. If you know that you should be there, that's great. If you think you should be there and you weren't invited, please let one of us know and we will take care of it. Peter, please go ahead.
Hi, thank you very much for having me. Yes, I'm just here to advertise that the Crick Institute in London is currently recruiting for early career group leaders in computational and theoretical biology. And so I'll just give you a short pitch about why it would be good to do. It's a really exciting and great place to work in London, in right near the centre and near lots of universities. And there are 120 multidisciplinary research groups there. And they're trying to really focus on recruiting some more theoretical and computational group leaders. There's close links to the Imperial College London, King's College London, and UCL, and it's also very close to other universities in the southeast of England. And so the group leader packages, they recruit group leaders for a 6-year contract, which is renewable once for a total of 12-year contract, and that comes with a core funding for 3 researchers and 2 graduate students, and an opportunity to expand if you want to apply for external grant funding. And there's lots of scope for internal and external collaboration. It's really set up to do really exciting collaborative science. So I would encourage everyone to apply. And we will send round a flyer to show when the deadlines and everything is. The deadline is 10th of March. So thanks very much.
So can I ask, what is the definition of early career?
I'm not entirely sure, to be honest. I'd imagine I wouldn't want to narrow it down, but there's lots of information on the website that I can point people to. I'll send a link over.
It's not quite the same. I had one of my graduate students has held a postdoctoral position in one of the research groups in computational biology at the Crick. Has been an amazing opportunity.
Yeah,
it's the opportunity to work with the best experimentalists in the world, the intellectual quality of the people he's engaged with. It's been tremendously valuable position for him. And of course, that's a postdoctoral position rather than a group leader position. But I don't suppose I have to sell people on the Crick. Everyone probably knows how good it is. But maybe not so much in computational biology. And I think his experience there as a computational biologist has been tremendously good for a place to build a career.
Yeah. It's fantastic. I'm a postdoc there at the moment, and yeah, it's fantastic, the ability to collaborate with so many different people, and there's so many different seminars and groups. It's really great.
Okay, thank you very much. You didn't have a screen share you wanted to do. We've got the link for the announcement in the chat, so if people
Yes,
want to
and
check
I've
that.
got a. I will send around. I think Lorenzo is going to be sending around a flyer, which which we have.
Great.
Thanks very much.
Thank you so much. Any other announcements before we get started? No. I want to say that next week we have 2 talks on the books, coupled systems that have in-host and between-host components, and mathematics in the quest for malaria eradication. March 3rd, we have a single talk from Alex Hoffman, modeling innate immune signaling, which should be exciting. We have opportunities for more people to speak on March 3rd. So far, we don't have anybody who's stepped up to speak on March 10th. So please help us fill the March 10th slot. Going forward, after March 10th, we have a number of exciting speakers in the works and lined up. Lorenzo can update us a little bit on the status of those things as we go forward, but please do help us for March 10th. That's currently open. So anybody who would like to have a discussion, pursue ideas, report on subgroups would be fun. All right. Other announcements or comments? Okay. Well, since we only have 1 talk today, the rules don't really have to be announced, so that's okay. I'm pleased to say that today we have Giuliano-Ferrari Gianlupi from here at Indiana University, who's going to be talking to us about his modeling work, and I will turn the floor over to him.
Thank you, James. Hello, everyone. We get started with the screen share here. Okay, so first, hello. Again, I'm Juliano Gianlupi. I work here at Indiana University at the Biocomplexity Institute with James Glazier, James Luca, and some other people that I'm sure are in the call. And today I'm gonna present on recent work that we did, asking ourselves, how can agent-based models improve the drug schedule studies? We wanted to know what are the effects of different drug dosing schedules on tissue outcomes. And there were a few reasons for that. First, we took Remdesivir as our muse drug, and Remdesivir didn't see widespread adoption. One of the reasons is that it is administered daily through an IV infusion that takes anywhere between 30 and 120 minutes. There are other reasons, like the animal models didn't translate that well to humans. But on the schedule side, if we could make the schedule be a dose every 3 days instead of every day, it could help patients that don't need to be hospitalized to actually stay on the treatment. We also wanted to ask how the effects of schedule change with different potencies. What other factors can affect efficacy? How can we apply agent-based models to improve clinical application of the drugs? Again, and we also wanted to ask something that can't really be done with a pure population and PK model, which is what are the effects of cell heterogeneity in their reaction to the antiviral on the treatment outcomes. So a pure population model wouldn't be able to do that because such effects would be washed out by the well-mixed conditions used in those models. And even if we add stochasticity to the model, like using a Gillespie solver, they would still be washed out by the mean behavior. And we come into this expecting that the infection progression will depend on the amount of time spent above or below the half maximum inhibitory concentration, IC50 for short. And again, we are using Remdesivir as a framing, but our questions are more general. And we are also using SARS-2 as a frame, but our questions are general to other viral infections, antiviral treatments, and even bacterial infection and treatments. And first, I'm gonna talk about our methods, and later on, I'm gonna go into our results. So to begin with, our agent-based model is 2.5D. meaning that we have a 2D plane where we have the epithelial cells. Here, blue are the uninfected cells, green, they are infected in the eclipse phase, red, they are releasing virus, and black, they are dead. And another 2D plane where we have our immune cells and diffusion of the virus, cytokines, and cytotoxic agents. Our agent-based model is composed of several submodels, one for each cell type, for the different diffusing agents, for immune recruitment, and intracellular replication. We calibrated our viral replication model such that the doubling time and decay rate of the viral load would match what we see in real life. And this was implemented in Computational 3D. We want to model treatment with an antiviral, and we are using Remdesivir as our muse. And so we have to include its pharmacokinetics. In reality, Remdesivir is a prodrug that is quickly metabolized inside the target cells into its active form. And Pfizer created Remdesivir in this way because its active form doesn't have good membrane permeability, which can be a good thing if you can manage to get the drug inside of the cells, because once it's inside of the cell, it's gonna stay there. And its prodrug form, on the other hand, has good membrane permeability. So it reaches the target tissue, gets inside the cells, and then the cells take care of making it active. And to begin with, for our pharmacokinetics model, we used a model from Gallo that is a full body PBPK model of Remdesivir, both in organs and intracellularly. And as we are focusing on the intracellular concentration in lung cells, we are able to reduce the multi-compartment model into a single compartment model. We calibrated our single compartment model to data from Humaniuk and the European Medicines Agency. And our single compartment model in blue here stays within a Gallo sensitivity analysis band in gray, and in black is Gallo's mean behavior in the sensitivity analysis. We made sure that our PK model would match the first peak of the dosing, and that's why it underpredicts afterwards. And we also had another reason to use a single compartment model, and that is that our cells in the agent-based model each have an independent copy of the pharmacokinetics model. And so having it be lightweight was pretty important for us so that our simulations could actually run in a reasonable time. And by having each of the cells have a different, a synchronized copy of the model, we are able to vary the parameters in each cell individually and investigate the intercell variability in drug response. We also, as I said, modeled the viral life cycle and the mode of action of the drug on that life cycle. And a virus in general to propagate has to enter target cells and then use the cell's machinery to unassemble itself, assemble new genome, more virus genome, generate more viral protein, assemble new virions, and then be released back to the environment. And we represent that by a series of ODEs. Remdesivir targets the RNA being made, the elongating RNA being made by RDRP. And that's an interesting step to target because that step has a exponential ramp up. So here we have the RNA quantity R, and we see that its rate of production is proportional to itself. So it ramps up exponentially and then saturates. The binding to the elongating RNA in our model translates to a modulation of this maximum replication rate, Rmax. And to find out a functional form for it, we fit in vitro inhibition data of remdesivir from Choi and Pizzorno, and found that a Hill equation, a decreasing Hill equation with a factor of 2 is a good functional form. Of course, we need to have also the IC50 for the drug in the cells. And we want to find the minimal dose that works and how that changes with different schedules and delays to treatment. And this is similar to a question that Goyal did in a previous study, but they did it in a pure population model. And we are adding space resolution and heterogeneity in the population. And the IC50 is easy enough to measure in vitro, but in vivo, inside of a patient's lungs, it becomes a bit complicated. And as a matter of fact, the intracellular IC50 of remdesivir is unknown. And again, we come into this expecting that the outcomes will depend a lot on the amount of time the system spends above or below the IC50. And therefore, to determine an IC50, we ran our PK simulation and chose the midpoint of peaks and troughs to be the IC50. We then explore different doses or potencies by numerically changing this calculated IC50 of 7.9 micromoles by multiplying it by 100-fold and 1/10 of its calculated value. And in clinical situations, drugs are usually given to achieve plasma concentrations 5 to 10 times the IC50. So we mostly focus on the 100-fold side of this variation. And as we change the schedule, we need to rescale the dose, of course. If we are dosing with 100 milligrams every day, and we go to once every 3 days, we have to then dose with 300 milligrams. And in the case of remdesivir, there's also a loading dose, which is set to be twice the regular dose. And for rescaling the loading dose, we actually have two options. We can keep it simple and set it to be twice the regular dose, or we can set it to be the regular dose plus the average over 24 hours. And we chose to set it to be twice whatever the regular dose is, and we'll explore the other option in future work. And as I said, we are adding heterogeneity in how the cells react to the antiviral in our model. And there are a few reasons that would happen in real life. There may be different availability of the drug to the cells, so effects from microdosimetry, especially if they have different distances from capillaries. The uptake rate may be different, especially if it depends on channels or pumps, which is not the case of remdesivir. But again, remdesivir is a muse more than the proper target. The conversion rate from prodrug to active metabolite will also depend on intracellular levels of enzymes, and cell age can affect metabolism. And on the enzyme point, we actually found some papers this week that point that this may actually be something seen with remdesivir, but I still haven't had time to explore those papers. And to explore each of these individually, we would need a complex model of the intracellular metabolic pathways of the prodrug into its active form. And to begin with, we are combining those into an effective change of the uptake or to the elimination rate of the drug. And we modulate those rates by drawing random numbers from a normal distribution and then multiplying the rate by 1 plus the random number. As we are trying to model treatment and figure out if that treatment is effective or ineffective, it's important that in the absence of treatment, the model sees a full sweep of the tissue by the infection, and all of our model's epithelial cells get infected and eventually die. In the original model, that wasn't guaranteed because we started the simulation with one initially infected cell. And in that case, we sometimes saw a failure of the infection to take hold. And that could happen due to viral stress of the single infected cell, or some immune cell got seated near it and killed the cell before it got to release any virus to the environment. And we found that if we start the simulation with 5 infected cells, we eliminate failures to infect and keep the medium time to full infection close to the original. And so on to results. With our initial investigation, we can already see 4 distinct cases of outcomes. I'm going to go over them in more detail later. And 2 of them are of effective treatment and 2 of ineffective treatment. And here in this figure, we see the several different treatment options. So going from left to right, we are spacing out the doses more and more from 24 hours, which is what we use clinically right now, all the way to once every 6 days. And from the top to bottom, we are steadily decreasing the potency or the dose that we are giving. And we see that for lower potencies, the treatment isn't effective, whereas for high potencies it is. And in the in the middle of them, we have some cases that are still effective, but not so much, and some cases where it is not effective. And as we go to higher and higher interdose periods, we see that the slow clearance cases creep up. And before I talk about slow clearance, I'm going to explain fast clearance, which is pretty straightforward. It's a type of effective treatment, and we classify simulations as fast clearance if the viral load goes to 0 before 14 days of treatment. There are no infected cells in the system, and there is no subsequent release of virus after it gets cleared. In the case of slow clearance, it's somewhat similar, but clearance happens after 14 days of treatment, or there is some clearance of the virus, but there still are infected cells in the system and they restart infection. And if their infection restarts, we still want to see the viral load trending down to classify it as fast clearance. And as a matter of fact here, for the dark blue band, which is our 0 to 100 quantile, we see that some simulations got down to 0 virus in the environment and then got back up. And we see the fluctuation trending down. The first of our ineffective treatments is persistent infection, which is a case where the viral load oscillates around a somewhat steady value, and the virus maybe have been cleared once, twice, or many times during the simulation, but it always restarts. And then widespread infection is also straightforward. We classify simulations as widespread infection if by 14 days of treatment, the uninfected population decreased by more than half, or if the uninfected population at the simulation end is less than 10 cells, or if the viral load is trending upwards at the end of the simulation. And here I want to point out some things in this viral load graph that we see a bit of the oscillation again, but in this case it's trending upwards. And we see a decrease of the viral load at the end, but that's because most of, if not all of the cells died already. So there's no more production of virus. And first, so effects of delaying treatment are not that major. Here we have the treatment starting with the infection, and here we delay it by 3 days. We see that there are more cases of slow clearance, and there is some creep up of widespread infection, but we only need to increase the dose by 15 to 25% to keep efficacy, which is not much. And as a matter of fact, there's a recent preprint by Pfizer from 3 weeks ago, give or take, that they say that even after the viral load peak is reached, if we start treatment after the viral load peak is reached, the treatment can still be effective. I need to read their preprint, but I'm not so sure. But on the matter of schedule, if our dose is 25 times the IC50, we can afford to dose every 3 days, even in the case that we are waiting 3 days to start treatment. And that translates to be about 2.5 times the half-life, which is quite large for Remdesivir. It's 30.5 hours. So maybe that's a possibility that could be explored in clinical trials. But this is all with the cells still acting homogeneously in regards to the antiviral. Once we add heterogeneity, outcomes in general become worse. So here on the left, we have the homogeneous case, and on the right, the heterogeneous case. And even when we start the treatment with the infection, we already see that more simulations got classified as ineffective treatments. And when we wait, then things become even more apparent. Waiting is even worse when we have heterogeneity in cell behavior in regards to the antiviral. And to be able to wait to dose every 3 days, in this case, we have to go all the way to 50 times the IC50 instead of 25 times the IC50. So we effectively have to double the dose as before, which can get into the territory of toxicity effects. And the reasons for that is that the cells that don't metabolize the antiviral well are responsible for most of the viral production. And by metabolizing well, I mean cells that are effective of taking the antiviral into its prodrug form inside of them and don't dump it out of them fast. And we see that here. We did some simulations where we varied either the uptake rate or the elimination rate to see this. And we see here at the top is the variations in uptake rate, and here the elimination rates. And we see that cells that take long to convert the drug into its active form produce most of the virus, whereas cells that are fast in doing that don't produce much. And the opposite is true for elimination. Cells that eliminate very slowly don't produce much, whereas cells that eliminate fast produce more of the virus. And there seems to be a plateau here and a linear relation here, although with the number of points we have in the scatter plot, things are not actually that clear. But again, that's all with a single amount of heterogeneity. We set the standard deviation in our modulation of the rates at 0.25. And but we don't actually know what are the levels of heterogeneity in a tissue. And so we changed this level of heterogeneity from 0.1 to 0.5. And we see that as we increase heterogeneity, we need to go into higher and higher required potencies to get the same effective results. And with the highest level of heterogeneity that we studied, barely any simulations got classified as fast clearance, only 6 out of the 110 situations we have here. And I mean, most of them were widespread infection, even for very high doses of 33 times the IC50, 50 times the IC50. And there's a simple reason for that. As we increase the metabolic heterogeneity, the antiviral levels in the cells become more and more dispersed. So here we have the homogeneous case, and we increase the heterogeneity for those 3 cases. And for the highest case, some cells simply don't accumulate any antiviral inside of them in our model. So that's our result. And for future work, we are going to improve our infection and immune models. We are going to include adaptive immunity, interference, antiviral activity, tissue recovery. We are going to include effect of drug toxicity. We are going to model other types of drugs, see how oral drugs would behave with our system. And we're going to add heterogeneity also in the viral production inside of the cells. And we are going to add heterogeneity in availability of the drug, which is something that we didn't explore here. And we are going to include a detailed model of the intracellular drug metabolism. So in conclusion, if the tissue cells metabolize the antiviral drug homogeneously, we can afford to dose with an interval of 2.5 times the half-life. And for a drug that requires AV infusion, that can improve clinical use. If you consider that the cells are heterogeneous in their behavior in regards to antiviral, we require higher doses, and in some cases they need to be way higher to afford to go to 22.5 times the half-life. We have to go to 50 times the IC50. This effects from heterogeneity wouldn't be able to be seen with a pure population and PK model, as those models are always well mixed. And so any heterogeneity would be washed out by the mean behavior of the population. And if we want to use models effectively in the design of clinical treatments, we have to quantify and use cell-to-cell heterogeneity in them. And we should integrate cell-to-cell heterogeneity when we build and apply PBPK models. And some things that I would like to ask your help with is actually quantifying the level of response heterogeneity in target cells of tissues. What is the distribution of that heterogeneity? Is it normal like we assumed? Is it uniform? Is it binomial, log normal, something else? And also in developing a in-depth model of the intracellular metabolism of the antiviral and finding which parameters in that model are the most sensitive to heterogeneity. And here I have my references, and that's all for me. Thank you for your time.
Okay, thank you very much. We have plenty of time for some questions. Ruchira.
Hi, thank you for the very nice talk and the very nice model. I really, really like this approach. You know, I had sort of thought of PK models as sort of inherently this continuous, you know, population level thing. So it's really, I think this will be powerful and can be used in many ways. The approach having these PK models in each cell. So I guess just one question, which is sort of following on to the last thing you said about the cell-to-cell heterogeneity. So as far as I understood, you mostly had heterogeneity due to the fact that, you know, cells are not identical, but it didn't seem to me that you had looked at the fact that there's different cell types, which could each be susceptible to the virus, but maybe at different levels. And so with that, you could also look at spatial structures. So I'll just name something that I'm a little bit familiar with is like an intestinal crypt has different kinds of cells in it. There's the enterocytes, and there's also the lymphoid tissue that is underneath. And so you could, and there's Paneth cells. So you can imagine that there's different types of cells, and so the distribution of the different types of cells, if they have different susceptibility to the drug, and they're also spatially arranged in a different way, that could also have some impact on the overall impact in the milieu. Would be based on that little ecosystem of these different cells.
Yeah, and in the lung, there also are different cells that are targeted by SARS-2, and that's actually an improvement that I forgot to add into the slides. But yeah, we plan on adding different cell types, and then try to see if there is an experimental data on how they may differ in their uptake or reaction to the antiviral, or even in the toxicity of the antiviral to different cell types. But yeah, that's definitely a improvement. that
Yeah,
need
and
to
as you said, there's many directions you can take this. So yeah, this is pretty awesome. Thank you.
Other questions? Lorenzo.
Hey, Juliano.
Hello.
Well, great talk. And I think this kind of model is very important because we know that drugs go in clinical trials and they fail.
Yep.
And we perhaps sometimes it's the dosing. Sometimes we want to stop them before, so we don't spend a lot of money, and so on and so forth. Regarding the heterogeneity, one thing that comes into my mind, perhaps have you checked? Perhaps I think there are around database on single-cell RNA-seq on lung epithelium. This might be giving you an index because I assume that there is one or two enzymes that actually metabolize that drug. Perhaps one on I'm not sure.
Yeah, I have. Where is that figure here? Yeah, there's a phosphoramylase, and according to Tarun, which is one of the authors, that's the rate limiting step in the process. So yeah, definitely, that's a good place to look for that type of information.
So if there is, what is the extent of heterogeneity in the expression of this particular gene?
Yep.
And well, my question was really more on the theoretical part. Let's say, all right, there is the assumption that a cell that doesn't metabolize well is unhealthy. And but these cells might also not be so good at producing virus because the whole machinery perhaps is turned down.
Yep. Yeah, and we completely randomized the uptake and elimination rate independently. Maybe we should have correlated the two. And that's something to keep in mind when we add the heterogeneity in viral production as well. Do it all independently and all correlated. That's a good point, yeah.
The other reverse thought is that it probably goes more in what you already done is that perhaps there is a physiological heterogeneity where a cell, for her own reason, keeps the enzyme down because doesn't need it or whatever, but then is very good at producing virus, and it will end up in the case that you mentioned. Well, just say that probably this is another switch that might be included in the model.
Yep. Yeah, thank you. Yeah, that's a good point. Yeah.
Thank you.
Other questions and comments? I feel awkward asking a question or a comment since I'm a co-author on the papers. I need to turn that over to other people today. I think if nobody will ask anything, I guess I'd like you to come back to your future work section. And maybe we could talk a little bit, and especially I'd like comments from people in the room about what this model that Juliano has presented is reasonably sophisticated, but clearly there are a lot of simplifications. And Lorenzo and Ruchira both made some suggestions about ways it could be made more realistic or significant. And I was wondering if there are other people's opinions about perhaps what the most important things to improve would be. What are the key, of all the things that you potentially you could add to it, where would you start in terms of trying to make it more specific? And also, I think, Lorenzo, your point was a good one. We looked and didn't see data on the phosphoramylase variability.
Yeah.
But that doesn't mean it's not there. It just means that our search terms were not right, or it wasn't findable through Google Scholar. And so it's definitely possible that there's a database that's sitting there where it's hiding. And there was a seminar a while ago where somebody said that repositories are where data go to die. because they're not findable. And so it's possible that in fact the data exists, but we're not finding them. But maybe it also suggests for other data sets that could be used to calibrate a model of this kind. If people in the room have suggestions, that would be appreciated.
Yep.
Well, I think it can be done experimentally in vitro.
Yep.
If some has a model of epithelial cells, I don't know if primary ones are culturable in vitro, but perhaps there are lines. And if there are physiological reasons for which they have alternating level of this enzyme, this might emerge from well, immunofluorescence or things like that can do. And in vitro, you then have the chance to do loads of acquisition. So you have the stats that are with you normally, and you can estimate probably the parameters. And then there is always the matter that people say not necessarily believe that this then is relevant in vivo, but then it's always is just one step forward and one after the other. Because then having an in vitro model, I think, is super, because one could also infect the cells then and actually validate, or can artificially create a model where there is heterogeneity by mixing cells that are not down or not for that particular gene, and then see if they have different ways and test some prediction in vitro. This could be, is the kind of thing. Well, as a cell biologist, I would think directly there.
Yeah, I also think that hopefully there is some in vitro data already, but if not, that's the place to go. And yeah, Ruchir also has a great point about cell cycle. Yeah, if the cell is at different stages, it's gonna behave differently. If it's right at mitosis, is more preoccupied with that than anything else, and may just kill itself with the drug.
Joy, was that a hand raised?
It was supposed to be, but I'm having computer problems, so.
Go ahead.
This may be what you're talking about when you say model heterogeneity and intracellular viral behavior. Just wondering if that includes viral evolution away from, it's an RNA virus, so it's going to mutate away from having the drug be effective at all. Which is, we've watched in the flu field for far too long. So just wondering if that's what you meant by that, or if that's something else you're throwing in.
No, that wasn't what I meant, but that's a great point to add.
Sorry.
No, that's something important to consider. No, I was meaning more that, well, if for whatever reason the cell has depleted its RDRP, or it has some mutation that it doesn't have as much DRP, it's going to produce less viral genome, for instance. But yeah, different viral lines. That would be an interesting one to add. Not sure how right now, but I'll think about it. Yeah, thank you.
Prego.
No, I think that's important. I mean, one thing here is that virus is modeled as a continuum.
Yep.
And so if you want to model viral strains, you have to decide how to represent those strain variations. And I think it's certainly possible to do it. And you could, but I think then you probably have to start modeling the virus as a set of objects rather than a continuum.
Yeah.
So just the way he's taken the background of the population of cells and made it individuals, you'd have to make the virus an agent as well. Of course, in this diagram, if the virus evolved around a restriction, then the functional dependence of the, say here, the replication rate R max as a function of the concentration of the drug would change. And so you could do that. And of course, some of the other, the new orally available antivirals that are now coming on the market, they don't attack the RNA-dependent RNA polymerase. They're attacking the protease that's dicing the viral genome. And so you could also think about modeling other stages in the viral life cycle. And we also heard that great talk by Kevin Janes the other day.
Mm-hmm.
And in this model, there isn't anything about having to coordinate the aspects of viral replication. It's a straight through process, and you don't have a situation which he was investigating, for example, where if you don't coordinate capsid production with RNA replication with non-structural RNAs and proteins, that you don't assemble effective infectious virions. And so you could definitely take a bigger model like that and use that, which would give you more directly meaningful drug candidates. And of course, he was actually looking at evolutionary questions. In that case, more on the host than on the virus, but still trying to connect those kinds of models to sequence data. So I think it's a great insight.
Yep.
Other questions today? People are quiet. Maybe the Juliano again. What's the next thing? What's the first thing you're going to change? What's your next project of that long list of possible projects to extend them all?
Oh, adding a proper model of the interferon and the adaptive immune response. And with the interferon, we will then be and proper adaptive immune response, we will be able to do combinatory treatment with interferon treatment and as well as antibody treatment, and combination of antiviral and all of those, and adding heterogeneity wherever I can think of that could have heterogeneity.
So one thing that this model does, and I think Ruchira alluded to it a little bit, is you have two components of the PBPK model, one of which is the transport of the drug in the body as a whole, and the other one is the metabolism of the drug inside the individual cells.
Yep.
And conceptually, those are two completely different layers, but mathematically, they have the same ODE structure.
Yep.
And in your model, you've actually collapsed the whole body layer inside of the cells.
Yep.
That sounds like a strange thing to do, although people who do heart modeling, for example, do that kind of thing all the time. They have a concept of an ion channel, but they don't have cells in their hearts. So that idea of collapsing layers when the intermediate's still present isn't as strange as it sounds. But of course, if you were going to try to do something like microdosimetry,
yep,
which affects the availability of drug outside the cells,
yep,
then that might require you to rebuild that larger scale model as a container.
Yeah, I would separate it in two probably. One controlling sort of the global availability of the drug, and then each cell taking from that into itself. And maybe it wouldn't even be global, because if I actually add in capillaries nearby the capillaries, the concentrations would be higher. I would probably actually have the drug diffuse, which is something that I didn't add here because diffusion of remdesivir was way quicker than the virus and cytokines. So it would put a numerical strain on the simulation that didn't seem worth it to begin with.
The drug goes by passive diffusion, or it has an active transporter or something?
Passive diffusion, as far as I know, but a lot of in the lungs there are a lot of capillaries, so everything should be pretty. Yeah.
My last one will be if you see, well, in my mind, this kind of the core of this model. So when a drug meets heterogeneity, might be applied. I'm not saying even, but might be applied to other disease.
Yep.
I'm thinking cancer because okay, there heterogeneity is a keyword essentially.
Yeah, Ruchira just asked about cancer. I think
sorry.
something similar has been done for cancer with microdosimetry and such, but I'm not sure if I'm not sure the level of that investigation, how exactly they added in heterogeneity and what type of heterogeneity, and if it compares to what I'm doing here.
Well, I think probably cancer is the place where people are the most sophisticated in terms of this kind of modeling, in terms of heterogeneity. Because there you have the disorganized vasculature that's typical of solid tumors as well, and so it's a pretty so there are a lot of factors that are different for normal tissue physiology. Jim, I wonder, I mean, you've of course worked on liver microdosimetry a lot, and also this putting metabolic models inside of cells. Do you have an insight into where you think whether the liver work could be informative here?
Yeah, I think it could. The difference is the liver is so much more active metabolically and zonated and all the other effects that are in the liver that I don't think the lung has.
Mm-hmm.
But nonetheless, I mean, the challenge is going from what gross measurements you have, which is usually dose and what's in the circulating blood, and trying to map that down into what's happening in an individual cell. That's always going to be really, really tricky.
Mm-hmm. Yep.
Okay, well, thank you. Any other questions for our speaker today?
I'll just throw out there that I've been looking recently into exosomes, and people look at them in terms of possible drug delivery vehicles, and they find that their uptake, they home to different tissues.
Mm-hmm.
So that's one particular type of, I mean, that's regardless of what's inside, they home to particular tissues differentially. So that's one thing that I guess could be modeled if that's the kind of drug. It's the delivery method itself. Does it have some inherent heterogeneity other than just receptors or proximity to capillaries?
Yeah, well, that's kind of already true with remdesivir, since it is a prodrug, and it has to be converted intracellularly. If the enzyme levels are different, then it's of the drug, not necessarily of anything else.
Right. So the actual drug itself ends up having tissue specificity because of the metabolism that's required to move the drug.
Well, I think your point, Richard, is, especially in something that's highly toxic, like a chemotherapeutic, targeting is critical. I think the claim initially was that Remdesivir was relatively low toxicity, although I guess some of these recent papers on why people don't use Remdesivir suggest that there may be more hepato and nephrological toxicity than they thought initially.
Yep.
I suppose any active molecule that's a drug is going to have toxicity somewhere. By definition, if it's active, it's going to do things you don't like. But I wonder if there's anything that's known about targeting in the lungs. It would be very interesting. I don't know anything about. I've read a certain amount about cancer targeting of various kinds, but I don't know anything about trying to target lung tissue specifically. That would be very interesting to know.
Well, normally for targeting lung tissue, you just use pulmonary delivery. Right, you use an inhaler.
Yep.
I suppose it also depends a little bit on what you want the drug to do. If the goal of the drug is to prevent viral spread within the lung, then inhalation delivery might be appealing. On the other hand, if you're giving it later in the infection and the goal is actually to prevent the virus from spreading between secondary sites of infection, then you might actually want it to be systemic.
Yep.
Okay, any last questions today? We've got time. We've talked out our hour. Anyone for a last question? Okay, well, I thank you, Juliano, for giving us a talk today. Very much appreciate people asking lively discussion. And we will see the steering committee tomorrow morning and everyone else next week. Thanks again for joining us.